Hello everyone, my name is Thomas and I would like to tell you more about what my team and I are working on at the Medical University of Vienna and at CEL. And in particular, we are interested in the role of non-hematopoietic structural cells in tissue immunity. Because let's be honest, every cell can be an immune cell. And as I said, we are in particular interested in how an immune response is shaped within a tissue. And we believe very much that it needs a tight interplay and also a controlled interplay of non-hematopoietic structural cells that perform an immune function that we call structural immunity, and how these cells are interacting with innate immune system and the adaptive immune system. When an immune response emerges, it not just suddenly appears, but usually it comes in different phases. Two examples of these phases are depicted here, and in those examples we can very nicely see the four phases of an immune response. It often starts at homeostasis, followed by the induction of an immune response. Next, we have the peak of inflammation, and then it comes to the resolution phase. And this is a very important point during an immune response, because here we can have two different outcomes. On the one hand, we can resolve the inflammation and the host goes back to homeostasis. On the other hand, if the immune response can't be resolved, we end up with chronic inflammation that can actually be detrimental to the host. Now, how do we model these phases and these different outcomes in the lab? We use LCMV viral infection models. And in particular, we use two different clones. One of them, clone 13, that results in a chronic inflammation, and the other one, clone Armstrong, that can be resolved and allows us to really nicely study the resolution phase during an immune response. Now that we have our model, I will quickly summarize what is the overall goal. As I said, we want to map tissue immunity over the course of acute and chronic inflammatory response. The way we do this, we use a viral infection model. And furthermore, due to collaboration with clinical researchers at the Vienna General Hospital, we also have access to patient samples that we can also profile. The way we do it is usually, especially from animal models, we take many different tissues to get a really good and global overview of what is going on in the host. And from each and every tissue, we isolate structural cells and hematopoietic cells. And then we profile those cells with orthogonal assays. So on the one hand, we use, for example, flow cytometry and immunofluorescent imaging. On the other hand, we also use bulk RNA sequencing and epigenome profiling using the ATAC-seq assay. And we perform single-cell RNA sequencing and spatial transcriptomics to really figure out what immune and structural cells are doing within tissues. With all our data, once collected, we perform integrated analysis where we take all the data from different assays as input. Then we construct networks for each assay, and then we can really start to compare our networks. For example, we can compare conditions, time points, and cell types to really dissect tissue immunity and figure out how structural cells and immune cells work together to mount an immune response within a tissue. And I think our research provides novel insights, as on the one hand, we can characterize structural cells at different phases of the immune response. Furthermore, we can deconstruct the interplay of structural cells and hematopoietic cells and really figure out, do they communicate different or similarly between tissues. We can also dissect regulatory network dynamics and identify key mediators that ultimately enforce structural cell immune function. And by this, we might also identify signaling and transcriptional regulators for therapeutic manipulation. If you're also excited about structural cells and structural immunity, just email me and get in touch and we can talk. Thanks.